All right, as we dive into our study of the cell, we're going to move into a lesson specifically on cell transport, how things move in and out of the cell. All right, and the main objectives we're going to cover in this lesson are something called cell differentiation. We're going to go over the big idea of the entire cell unit, and then, like I said, we're going to dive into cell transport. Before we do dive into cell transport, I want to make sure that we have a good understanding on what you covered in the types of cells activity, where you went through and differentiated between eukaryotic cells and prokaryotic cells, and then plant and animal cells. So we have different cell types. All right, and as we get this started, we should make note that the cell is the basic building block of all life. So for those of you that go on and become doctors, you're going to be getting a biology degree, and you'll take entire courses on just the cell, and rightfully so. We would not have living organisms if we did not have this thing that we call the cell. So the two main cell types, I'm going to break this down for you in kind of a flowchart manner. So the first type is going to be what we call prokaryotic cells. And you should have gone through and made a T-chart and Venn diagrams for these already. So I'm not going to get into a lot of detail. I'm just kind of doing a quick overview and a review of it. Eukaryotic cells then are our other group of cells. All right, so all cells on Earth are going to fit into one of these two categories. All right. You guys know I like to look at the literal meaning of a word, and prokaryotic, pro is going to mean before, karyo is going to mean kernel, all right, or basically nucleus, so before a nucleus. Eukaryo is going to basically mean that there is a kernel present, all right, or a nucleus present. Or with nucleus would be or with kernel would be the most specific explanation of that. And then underneath our eukaryotic cells, we're going to break off then into both plant and animal cells. From the last lesson that you completed independently, you should be starting to identify the organelles that make up each one of these types of cells, and understanding that there are certain organelles that are specific to plant cells and certain organelles that are specific to animal cells. But we'll be diving into that in more detail as we go forward. Prokaryotic cells are also going to be broken up into two groups or two distinct domains. The first domain would be Archaeobacteria, and our second domain, and this is going to be the domain that covers the traditional bacteria that we are used to. This is the closest thing to eukaryotic cells, but obviously they're still in the prokaryotic group. Here would be eubacteria. All right, the Archaeobacteria, these are going to be things that we find in extreme parts of the world, extreme environments with really high temperature, high salt concentration, a very unique pH, like hydrothermal vents at the bottom of the ocean, places like that. The eubacteria, this is going to be your traditional bacteria, things like E. coli that you might find on your kitchen counter even, right? You could find E. coli from a piece of old meat, something like that. And again, these are going to be the type, these Archaeobacteria that survive in extreme environments. All right, so make sure you have that down. Again, a nice easy differentiation between the different cell types, prokaryotic, eukaryotic. The prokaryotic being the Archaeobacteria, and the eubacteria and then the eukaryotic side being plant and animal cells. And we're going to be diving in, obviously, in this unit, learning all about those as well. Now we're going to move into what we call cell differentiation, our first main point of this lesson. So how a cell differentiates is very important. Differentiation. Before we can really dive into differentiation, we need to talk about the emergent properties of cells. Emergent properties, basically, like I said here, they happen when we have an interaction of individual components. We kind of see this from the endosymbiotic theory at the beginning, but the idea from Aristotle that the whole is greater than the sum of its parts, right? So you're going to have the individual organelles that are going to work together to create the overall function of that individual cell. So let's go ahead and add that we have the individual organelles work together to execute the overall function of the cell. So as we start to learn the functions of the individual organelles, we'll see that each one is an important part to the overall function of that cell. All right, good. So once you have those things down, once you understand the idea that emergent properties, or the idea really that cells, individual cells, are going to be working together, now we can dive into the idea of cell differentiation. Okay, so for cell differentiation now, first of all, what is it? So we're going to be able to connect the ideas of cell differentiation with the idea of emergent properties here in a moment. But first, let's talk about what cell differentiation is. Okay, it's basically the ability of certain cell types. I want you to take a moment and try and think about a type of cell that you've heard that is very powerful. There's a lot of research being done on this type of cell. Try and think of what that cell type would be, and then we'll come right back. Hopefully you had a moment to kind of brainstorm. So this type of cell is what we call an embryonic stem cell, which I'm sure you've heard of. Embryonic stem cells are unique because they can differentiate. All right, they have the ability of certain cell types to change into other cell types. All right, and now this is what will lead us into our idea of emergent properties. Okay, so cell differentiation is the ability of certain cell types to change into other cell types. Basically, these are what we call our embryonic stem cells. And embryonic stem cells are, this will be a new word for us, pluripotent. Pluripotent. All right, and pluripotent basically means it's a cell that has the ability to differentiate. Okay, so let's take a look here now. This really is our big idea for this entire unit. The big idea is the idea that we're going to have cells that are able to have emergent properties, and they are also going to be that we have cells that can differentiate. So we start with embryonic stem cells first. So these embryonic stem cells, they can differentiate into a bunch of other different types of cells. For example, heart cells would be cardiac cells. So the embryonic stem cell is going to be in the embryo of a new animal. Okay, and those individual embryonic cells are going to differentiate into all of the different cell types that make up an organism. So we have cardiac cells. We could have digestive cells. Right? We could have skin cells. Okay, let's take the digestion cells and use those as our example. All right, so we go from, again, embryonic cells. We'll use our digestive cells as our example. So if you get a bunch of digestive system cells together, all right, so the embryonic stem cells have programmed, and they are going to become digestion cells. Now we're going to have digestive tissue, okay, which is the next step up from a digestion cell. So right here I have my individual digestion cell. Same thing would happen with a cardiac cell. I'd have some individual cardiac tissue, right? Okay. Now then we're going to move from digestive tissue. We get a bunch of digestive tissue together, and that is next going to form a digestive organ, such as maybe the stomach, all right, or the large intestine, perhaps, all right. In the cardiovascular system, we'd have a cardiac organ, like the heart, all right. And then we move on from a digestive organ to the actual digestive system, okay, which is going to include the stomach, the small and large intestine. So it should make sense when we talk about emergent properties here, how we can go, we're emerging from the digestive cell. We get digestive cells together, it's going to make digestive tissue, digestive organ, which would be the stomach, and then the digestive system. And then from there, like up here, we would have the heart and the blood vessels. All right, same thing with the skin. We'd go through this same pathway or process, and then we're going to get to an actual organism as we go. So we've got all of the cells mixed together to make tissue, and then the organs, and then the organ system, and then you put all those organ systems together, and we've got an organism. But the big idea here is that this started all the way back with the embryonic stem cells in the embryo of that organism early on in life, right? That embryo then is going to differentiate into these different types of cells: heart cells, digestive cells, skin cell, muscle cell, all right, the liver cells, all of the different organs, okay? It's going to form all of the different organ systems of the body, and then eventually we get to the miracle of an organism. All right, so moving on now, we're going to dive into cell transport. What is cell transport, you might ask? Well, we're looking at just how things, specifically things, solutes such as sugar, salt, or maybe some waste in the cell, how do they move in and out of the cell? Okay, so that is the primary focus of what we're getting into here today. So there's going to be two types of cell transport. So as we look at cell transport, we're going to focus on something that we call ATP. ATP is short for adenosine triphosphate. This is the energy currency of a cell. So, for example, you go to a store to purchase something. You can't just tell the clerk at the store, hey, I have money in my checking account, and then walk out of the door, right? You need some form of currency. So that's what we're looking at here. This energy currency is the energy that's going to help to complete all of the processes of the cell. So eventually it's going to help to complete all of the processes of an organism, right? Since we talked about emergent properties that go from an individual cell to an organism. ATP is made in the mitochondria, and it's made via the process of cell respiration. All right, we're going to be getting into cell respiration very soon in this class as well. All right, so with ATP being the big idea here in terms of cell transport, now we're going to take a look at the two types of cell transport. All right, the first type is going to be, there's going to be two types, and they're both going to have to do with ATP. The first type is going to require ATP. The second type is not going to require ATP. There's no ATP involved. Okay, so let's take a look at the first type. This is what we call active transport. All right, it requires some form of energy. Okay, and this is going to be the movement of solute up the concentration gradient and that's going to require ATP. So we'll say via ATP. All right, we're going to take a look at a quick little video clip here that's going to really start to help us get an idea of what a concentration gradient is and also what diffusion is.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy.
So this should make sense, right? That if I were to dump, for example, some Kool-Aid powder into a a beaker here of water, those molecules, they're going to kind of spread out evenly, right? They're going to move from an area of high concentration to an area of low concentration just kind of naturally.
One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water.
This is a concept, guys, in biology that's really commonsensical that you've seen over and over and over throughout the course of your life. So now I want you to take the time to focus in on the specific reasons why these molecules spread out evenly.
What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called.
So guys, make sure you write this down. We'll be going over this in more detail, but this is the concentration gradient that we're talking about, moving from high concentration to low concentration or low concentration to high concentration. So make sure you get this down, and then we'll go over it together.
Called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
Again, this is something that you've seen naturally. I gave you the example of putting some Kool-Aid in some water and seeing the Kool-Aid spread out evenly. So it's nothing that you've never witnessed before, but now we're just learning the specific science behind it. Concentration gradient, I want you to think of it as a gradient, kind of like a hill, right? Hill and gradient are synonymous with each other. So that's what we're looking at. So if we say that something has a high concentration, then it's going to be higher on the concentration gradient. It's not actually a hill, but you're just going to see that term, concentration gradient, a whole bunch. So I want to make sure that you are very familiar with it, all right? So if we say that up here we have this many molecules of Kool-Aid, and down here we have less Kool-Aid, then obviously we're going to assume that the Kool-Aid molecules are going to naturally move in this direction. If one of my students is sitting at the top of the steps and I push him down the steps, probably not a good idea, but it would be easier for that student to go down the steps, right? It's going to be easier for that student to go down the steps than to go up the steps. And then this is the idea of where energy, or ATP, come into play. All right, so to start with, though, understand that the gradient relates to the slope, okay, or a hill, that kind of thing is what we're looking at. So naturally solutes like salt, sugar, are going to be able to move down the concentration gradient through simple diffusion, right, requiring no energy. They just go down the concentration gradient. Okay, and let's go back now to our different groupings here. So far we've got that cell transport. What is it? It's how solutes such as sugar, salt, waste are going to move in and out of a cell. And from our video, we started to look at what the word diffusion means. And we are on active transport, which is the movement of a solute up the concentration gradient via adenosine triphosphate, or this ATP. Right, so on the one side, we've got our active transport, which is what's going to require ATP. And then on the other side, we have just regular diffusion, which is going to require no ATP whatsoever. We're going from an area of high concentration to low concentration, and overall we're going to call this passive transport because it does not require ATP. There's going to be two types of passive transport that we're going to look at. The first one is simple diffusion. We saw that in the video. We saw an example of molecules or a solute. In that case, it was sugar. Move from an area of high concentration to low concentration. All right, so for simple diffusion, go and get this down that it is the movement. So this is again, guys, this is the fancy, sciencey definition of diffusion, but really diffusion is very straightforward. The movement of a solute. So when we say solute, we're saying something that dissolves in a solvent, like Kool-Aid powder. Something that dissolves in a solvent like water, okay, from high concentration to low concentration, down that concentration gradient that we just discussed. Okay, so what does that look like? We're looking at something like this. Again, we have a high concentration. We'll say that this is sugar. It's naturally going to move to the area of low concentration. All right. We have another type of diffusion, and do not get confused with the fact that the word facilitated is in this type of diffusion. So this diffusion is going to be called facilitated diffusion, but it is still diffusion. It does not require any form of energy. So go ahead and get this down that, again, it's diffusion. So it's still movement from high concentration to low concentration. It's just going to be something that's a little too large to go through the cell membrane by itself. So it's going to require some form of a channel to help it move through the cell membrane. I'll show you a little more specifically how it works. Let's take a look at an animation here of facilitated diffusion. You can see here on the outside of the cell, we've got these large molecules. We'll say they're glucose or sugar molecules, and they're just a little bit too big to go right through the phospholipid bilayer of the cell membrane, so they require a channel. Now, this channel is not going to be using any form of energy for this to happen. That's important. It's still passive transport.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
From all of this now, you'll be able to complete the stop frame animation assignment and upload those. That would be the next thing. Now that we've completed our lecture on cell transport, cell differentiation, and the emergent properties of cells.